Very good afternoon to all the participants. Thank you for joining today's webinar. We are very excited to partner with Amar Biosystems to host this session on how you can synthesize nanoparticles with microfluidics and contribute towards advanced drug delivery technologies. We have with us Kirti from Amar Biosystems, who is going to discuss more about their platform Nanomake, which is an automated nanoparticle synthesis platform. We also have a guest lecture by Dr. Swetha Raghavan from NCBS, who's going to talk about mRNA vaccines and mRNA therapeutics uh, with the perspective of applications in dengue intervention. Before we begin the session, let me introduce Venture Studio. Uh, I just want to briefly show you about who we are and what we do and who we serve. So Venture Studio is one of the oldest and leading technology business incubators in Ahmedabad. And since 2011, that was when it started with a collaboration with the Stanford University's Design School. And since then, uh, Venture Studio has supported more than 170 startups in different domains. We are a sector agnostic incubator, and more than 50 different products have been developed by our startups in different areas, including healthcare and diagnostics, medical devices, industry, manufacturing, fintech, hardware, AI, and many other domains. So our primary verticals include funding, facilities, and mentorship. We provide milestone-based funding through various government grants to startups, primarily in the early stages. And we have three different facilities that I'm going to talk about in the next few slides. And we also offer mentoring support in the form of business development, uh, finance, legal, patents and trademarks, as well as team building and marketing strategies. We have a lot of ecosystem engagement activities that go on. You can check about all our latest events and programs on social media. And I'll be sharing the details about our website also in the coming slides. So we do a lot of events, standalone and in partnership with different entities of the ecosystem to provide a strong 360-degree support to our startup community. We have the co-working and the events hub in the North Campus, whereas in the Central Campus, we have two specialized facilities. One is the DST Prayashala, which is for prototype development, and other is the Bionest facility, which is specially designed to support the biotech startups. So our Prayashala includes a lot of equipment related to prototype development, such as the PCB prototyping machine, your 3D printers, VMCs, laser cutters, and you have a suite of design softwares. So you have a design cell where you can do uh, modeling of your product, and the Bioness Lab facility is designed to support startups in the biotech domain, where we provide them workspace, equipments, technical support, as well as mentorship. You have a lot of equipments and facilities, including your basic instrumentation, and then we have workspaces. You have meeting rooms and collaborative workspace. You have the microbiology labs, you have chromatography labs, we have molecular biology setup. We have a BSL2 cell culture lab where you can carry out your cell based assays for mammalian cells and tissues. We have a lot of other dedicated facilities like walk-in cold chambers and freeze dryers. And if you want to apply, the process is very simple. Anybody who's interested in using our facilities, whether it is Prayashala or Bionist or our co-working space, you just simply write an email to us bionist@ahju.edu.in with your requirements, and we'll get back to you at the earliest. These are some of our startups, our users who have been using our facility. And if you want to reach out to us, we are also active on social media. We are present on LinkedIn and Instagram and YouTube. And you can also check out our website, venturestudioindia.com, to know more about our services and facilities. So once again, I thank everyone for joining this session today. We have an exciting session ahead of us, and I'm going to uh, request Dr. Alazar to take over the session. I will stop the screen sharing now. So we have Dr. Alazar from Amar Biosystems.
Hi, Albert. Thank you so much for that introduction.
I think there is an echo from your end.
Yeah, is it better now?
Yes, yes. This is fine now.
Yeah. So, hi everyone. Good afternoon. Thank you so much for joining us for our very first webinar series in association with Ahmedabad University Venture Studio. We are glad to welcome you all, and I would quickly like to introduce myself. I'm Dr. Alazar Kulumbala, and I take care of the business development and sales here at Amar Biosystems. So for today's presentation, we have our formulation scientist who handles the entire R&D at our facility, Ms. Kirti Ranganathan. She'll be walking us through a quick presentation, giving you an overview of the company, and certain highlights on microfluidic devices, lipid nanoparticles, and our very own product, Nanomake-L, and how that has led to bringing us a technology that is at par with the foreign alternatives. So I'll hand it over to Kirti, and she can take it from here. Kirti?
Yes. Thank you, Azar. I'll just share my screen. And hello. I hope the screen is visible.
Yes,
It
if
is,
you
right?
can
Yeah.
just put it on presenter. Yes, now it's full screen.
Okay. So I'll just and begin. So firstly, thank you so much, Dr. Arpit, for arranging this session. And so I'm Kirti. I work at Amar Biosystems. I'm a research scientist here. So I'll just begin a little bit about our startup. So we are a startup based out of Mumbai, and we specialize in microreactors and microfluidics based technologies and solutions for the biopharmaceutical industry. And currently, we are developing a microfluidic platform for the preparation of different types of nanoformulations. And so I'll briefly start a little bit about drug delivery systems, then introduce nanoparticles in drug delivery, and then discuss the different methods of preparation of these nanoparticles. And then we'll segue into microfluidic space synthesis. And finally, we will have a short product demonstration of our automated microfluidic platform. So, for audiences that are not really from this field, a drug delivery system is essentially a carrier material that allows you to deliver your therapeutic agent to a specific site in the body. So, conventional ways of drug delivery often face several limitations, such as poor bioavailability of the drug, poor absorption of the drug, and there could also be challenges with systemic toxicities. And a drug delivery system is basically a strategy, it's a formulation that allows you to deliver a therapeutic agent at the right site, at the right time, and in the right quantity. And the overall goal of a drug delivery system is to improve efficacy, minimize side effects, and enhance patient compliances. And we have different kinds of these drug delivery systems, such as your liposomes, emulsions, uh, other kinds of nanospheres, and polymeric particles. With that introduction, we also need to talk about nanoparticles. So these are particles in the size ranges of about 1 to 1000 nanometers, and they can encapsulate different kinds of drugs, biologic materials. And based on the type of the carrier material, we have different types of these particles. And in today's discussion, we would largely be focusing on lipid nanoparticles. Now, these are lipid based particles, but there are also different other types of particles, such as polymers, inorganic materials, and even complexes between lipids and polymers, and different kinds of hybrid kind of formulation strategies are also available. And some of the advantages of these nanoparticle based systems is basically they overcome the challenges faced in conventional ways of delivering drugs. So this particulate system will ensure there's more bioavailability, there is better encapsulation, better solubility, and less side effects. And as the advantages outweigh, we can see that there are many FDA approved nanoparticle based products that are currently available in the market. And some of these products are largely used in the treatment of cancer, infectious diseases, rare diseases, and some of them also go in vaccines. And one of the most recent examples would be our COVID-19 vaccine. That was a mRNA lipid nanoparticle, and with that introduction, we can also talk about lipid nanoparticles in general. So these are nanoparticles, and the size ranges of about 50 to 100 nanometers, and they are composed of different compositions of lipids. Now, each of these lipids have a specific role in the formation of the nanoparticle as well as in its in vitro activity. So the nanoparticle is composed of an ionizable lipid. This is a charged molecule that essentially encapsulates, electrostatically assembles with your nucleic acid, which is your actual therapeutic agent. We also have pegylated lipids, phospholipids, and sterol and each of these lipids have their own functions. Some of them form the bilayers of the nanoparticles, some help in preventing aggregation, and overall, this nanoparticular structure encapsulates your cargo, which is your nucleic acid, or any other kind of therapeutic molecule. And we have several different quality attributes, especially associated with these nanoparticles, that determine its efficacy, which could be a nanoparticle's morphology, its structure, its particle size, size distribution, its encapsulation efficiency. So these are some key quality attributes that need to be monitored, characterized to ensure that your drug delivery vehicle, which is your nanoparticle, is going to have the right activity in vitro. So that brings us to the question, how do we prepare these particles? So there are different ways of preparation of these lipid nanoparticles, and some of the ways are commonly listed, such as your ethanol injection, pipette mixing, vortexing, thin film hydration. So these are methods in which your aqueous phase is injected into ethanolic phase. And there are different mixing technologies, and some of them are largely used in lab-based preparations, and they're quite simple and easy to use. And in case of thin film hydration, we see that these technologies are largely used in preparation of liposomes. Now, most of these techniques that are listed are batch based techniques, which means that there is a challenge in getting the kind of reproducibility which lacks in these kinds of technologies. So, due to uncontrolled mixing, there could be inconsistencies between different batches of lipid nanoparticles that we prepare. And to circumvent all of these problems, we have a newer technology, a space that we specialize in, which is basically microfluidics. And in microfluidic technology, we essentially have, as you can see in the image, we essentially have a, let's get the laser right. As you can see in microfluidic technology, we have a microfluidic device. Now, these devices are basically have micron-sized channels, and these micron-sized channels will cause liquid phases, that is your aqueous phase with your buffer and your ethanolic phase with your lipid, will be injected into this microfluidic chip. And due to rapid mixing within these microchannels, there is better control over particle size and uniformity, and hence your lipid nanoparticle formulations are more precise, more reproducible, and also more scalable. And with this, I'd like to talk about microfluidics in a little bit more depth. So, microfluidics is essentially manipulating fluids at the micron scale to precisely design channels, and this enables continuous, controlled, and rapid mixing between different phases. So, microfluidic technology can also be leveraged for preparation of other types of particles that are not just lipid nanoparticles. We can prepare polymeric nanoparticles, we can prepare emulsions, and any kind of miscible or immiscible phases can be mixed using these micron sized channels. And the way the device works is you can adjust the flow rate, flow ratios, and modify the channel geometry to get particles of your desired size and encapsulation efficiency. So this is, in essence, the principle behind microfluidics. And as you can see, we can compare batch-based methods such as your ethanol injection or your pipetting with microfluidic-based methods, and we can see that in terms of variation between batches or reproducibility or better control over particle size, microfluidic methods always are preferred. And in terms of even droplet generation, and there's also another advantage is the reagent distribution and the consumption of reagents. So with very less amount of reagents you can get a better yield of your particles as well. So in order to understand how lipid nanoparticles can be prepared using a microfluidic platform, we need to first define the end goal or objective of our experiment, which would be to get a desired particle size and a desired encapsulation efficiency. So, in general, a workflow would be you prepare your lipid mix, you prepare your nucleic acid solution, you use the microfluidic platform to prepare the lipid nanoparticle, and after downstreaming, you would characterize it. Now, with an idea of what kind of particle sizes you're aiming at and what kind of encapsulation or what is the lipid composition, so these criteria help us decide the kind of microfluidic platforms that we can choose. Now, in general, we have different types of these microchannel device, microfluidic devices with a lot of microchannel geometries available. So we have simple geometries like a Y mixer, a T mixer. Now these kinds of mixing devices essentially uh, mix fluids through. So when two fluids are injected into the devices, there is laminar flow, and all the mixing is happening due to molecular diffusion. In comparison to these kinds of simple mixers, we also have advanced mixers like a herringbone shaped design, which would allow better mixing, and a flow focusing based device. So these are also microfluidic devices that are available currently with us. And in essence, another important aspect that we can discuss is also the choice of material. So as you can see in the left, this is a very simple PDMS based microfluidic device. There's a microscopic slide and a PDMS based device attached to it. But in essence, we can also prepare these microfluidic devices with different types of materials based on our applications. So we have devices made out of glass, stainless steel. Now, these materials are more compatible and with different kinds of solvents, and they can also be sterilized. On the other hand, we also have other kinds of materials like COC, COP, which are basically thermoplastics. We also have PDMS, which is popularly used in lab scale microfluidic experiments. And at Amar, we also have Teflon based devices. Now, these allow for rapid prototyping. And the cost also of these COC Teflon devices is quite lesser than comparison to glass or stainless steel. So once these parameters are set, then we have to optimize the formulation composition. So in the table listed, you can see a typical formulation composition for preparation of a lipid nanoparticle. And within a microfluidic setup, there are mainly two process parameters that we can optimize, which would be the total flow rate and the flow rate ratio. So the total flow rate will govern the mixing time and hence influence the formation of the nanoparticles. And it's quite understandable that at a higher flow rate there is faster mixing, and this would lead to smaller lipid nanoparticles. And similarly, flow rate ratio is essentially the controls the ratio between the solvent phase, that is the organic phase, to the aqueous phase, and this also directly impacts the particle size and its encapsulation efficiency. And as mentioned earlier, different quality attributes of these particles can be characterized using different methods, such as particle size and polydispersity index can be determined using DLS. Your encapsulation efficiency can be determined through various types of assays, as well as the morphology and functional potency. And as you can see in the graph, we have used a microfluidic process to optimize the particle sizes. So as we can see, with increasing flow rates, the particle size decreases. And this is only one parameter that we can optimize. In a sense, there are multiple parameters that can actually be influencing your particle size and your encapsulation, such as the microchannel design, the solvent type, the flow ratio, and so on. So that brings me into another interesting topic, which is automation and microfluidics. So conventionally in lab scale setups, we notice that we use manual syringe pumps, which are operated manually, and it's quite laborious to operate. So in the flow chart or schematic, you can see a setup for a double emulsion preparation. So there is a microreactor. There are two microreactors with three syringe pumps injecting three different fluid phases, and hence the emulsion is getting collected here. Now, operating such a system in conventional microfluidics is quite laborious, and there could be discrepancies in sample collection, the time that you press the syringe pumps, and these challenges are something that are quite popular and something that everybody faces who works in this space. Apart from that, microfluidics also has some inherent challenges with microchannel clogging, material compatibility, and scaling up. Since the process is restricted in micron sized channels, there are only a few strategies that people can use to scale up. It could be either having parallel chips or multi channel chips that could allow you to scale up. But what we are trying to do here is automate the entire system. So that could lead to a platform that will enable you to perform all of your nano formulation preparation at the touch of a button. So the user has to just prepare their formulation components, load it on the system, enter the parameters, and the system does all the work for you, from the preparation to sample collection as well as cleaning. So this ensures that your system is ready for the next formulation run, and there is no chance of microchannel clogging because the system automatically cleans itself. And this actually helps in faster optimizations It's more efficient process, and it also reduces operator dependencies. And based on this, I'd like to introduce our product, which is Nanomake-L. It's India's first automated microfluidic platform for lipid nanoparticle synthesis. And this platform is a benchtop unit. It's user friendly, and it allows for rapid optimizations and of nano formulations. And this system comes with reusable microfluidic chips, and it has an inbuilt wash program that allows you to clean the system after every formulation run. And the system is perfect for R&D scale optimizations of sample volumes from about a few microliters to about 20 ml. So we have generated some data on Nanomake-L. So here you can see we are optimizing our lipid nanoparticle preparation on Nanomake-L. And this is our microfluidic chip. As I mentioned earlier, it's a Teflon-based chip. So these chips are more compatible with solvents. They are clog resistant because Teflon is a non-sticky material in comparison to glass or COC, and we prepare these nanoparticles on the system. And the geometry of our device is essentially a split and recombination type micromixer. And this design allows for increased residence time within the microfluidic chip, but it does not cause microchannel clogging in comparison to a herringbone shaped design. Now, we achieved a particle size of about 85 nanometer and an encapsulation efficiency of more than 85% for plasmid DNA and more than 90% for mRNA. Similarly, we have also optimized this system for preparation of polymeric microspheres. And here you can see we achieved a particle size of about 50 microns and a variation of less than 10% between each batches. Similarly, we have also extended the application of the system for preparation of liposomes. Now, these liposomes range from particle sizes of about 54 nanometer to about 131 nanometer with very low PDIs and high encapsulation efficiency for proteins. So that is a general gist of Nanomake-L and its applications, how it can be used, and how it can be integrated into your workflows for preparation of different nanoparticles. And with that, I would like to just show you a product demonstration video, and that should summarize everything that we've discussed so far. The demonstration video will basically highlight what a typical formulation run looks like on the system. So I'll just play the video.
I think the audio is not coming. We have the audio. The audio is not playing. Kirti, there's no audio coming in the video.
Uh, just a second. I'll just try to fix that.
You can also play the video out of the presentation and share the video. I think you can then share the audio also.
Um, so I think
You
I'll
have
just
you have
disconnect.
a video on your system outside.
I'll just
rapid optimization of nanoformulation. My name is Kirti and I'm from Amar Biosystems, and today I will be demonstrating our microfluidic platform Nanomake-L. Nanomake-L is an automated microfluidic platform used for rapid optimization of nanoformulations such as mRNA.
Also, I think if you guys have a link to a YouTube video or something for the same, you can also share the link in the chat box so that anybody who has missed out on the video can check it out later. Meanwhile, I think Kirti, you can just try to just play the video through your media player uh, rather than using PowerPoint.
Hi everyone, my name is Kirti and I'm from Amar Biosystems, and today I will be demonstrating our microfluidic platform Nanomake.
I think you can share that browser screen. I think it was playing fine.
Nanomake-L. Nanomake-L is an automated microfluidic platform used for rapid optimization of nanoformulations such as mRNA lipid nanoparticles, drug-loaded nanoparticles, polymeric nanoparticles, and emulsions for applications in vaccine development and cell and gene therapies. Let me walk you through a demonstration of our equipment. Here we have three syringe pumps, P1, P2, and P3. This is our microfluidic chip. This is the fraction collector for collecting waste and sample, and this is the wash pump. To operate the system, we use this interface. First, we have to select pump and press pump 1 and 2. Once they turn green, we need to initialize this pump. Initialization ensures that your syringe is back in its home position. Once the pumps have been initialized, then we need to load our precursors into the tube holder. Pump 1 has our lipid mix and pump 2 has mRNA in citrate buffer. Once they have been initialized, we go back to the home screen and enter run. The run window has different parameters that can be optimized for each formulation. We have already pre-entered the values such as the total flow rate, the flow ratio, the sample collection volume, and the initial and end waste collection volume. Once you have entered these values, you can select load value and review the run. Once the run has been reviewed, you select start and start process. Now the syringes are being loaded with our precursors. Once the syringes have been loaded, they will be injected into the microfluidic device and the initial waste and the sample will be collected. Once the formulation run is over, the system wash function will be automatically switched on. This is provided to ensure that the microfluidic chip can be cleaned after each formulation run and hence can be reused. Once the system wash function is done, the fraction collector will move back to its home position and you can collect your sample. So now the formulation run is over and you can collect your mRNA LNPs. This sample will be further characterized for particle size, polydispersity and encapsulation efficiency.
If the voice is not audible,
Yeah, is it audible now?
yes.
Because I get muted every now and then. I'm
No,
not.
no problem. It's audible now. It's audible.
Yeah. So yes, as I was saying, our entire team is on the is in the webinar right now. So if you have any queries or any questions, we'd be happy to answer, and you can reach out to us for scheduling a demo of the product or any kind of services that we could provide. We'd be happy to discuss. Thank you so much for your time.
Yeah, thank you, Kirti, so much. We have a few questions that we can take
Mm-hmm.
in the chat box. So there's a question from Arya Agarwal about how are the particles controlled in the microfluidic device? Are these devices purely polymeric in nature or do they also have electrodes? Because it seems lipid nanoparticles do have a charge. So I think the question is related to how the particles are moving, how the different solutions are moving in the system? I think that's what the question is.
Um, so basically, the microfluidic device has tiny channels. Now, these are micron sized channels. And when you inject fluid into these tiny channels, there is better control over the mixing that is happening inside these channels. And that leads to controlled nanoparticle formation. And that helps in maintaining a constant particle size. And added to the next question of the microfluidic device, so as I mentioned, there are different materials that can be used in the fabrication of these devices. Some of the materials are plastics that are polymeric, but they're also glass based devices, silicon based devices. So there are a lot of varieties and options, and you choose a device based on your application.
Okay. Another question by the same person is that are the chips prepared in-house or are they outsourced?
Um, so the chip that we have, so currently we have a Teflon based chip which is prepared in house. So the designing, the fabrication, the assembly, all of that happens in house currently. And we have a Teflon based device, but we're also developing COC and COP based devices, and silicon based is also available.
Okay. There is one more question from Dr. Parth, who is asking as to if we can implement this method for herbal medicine or herbal extracts.
So, if you want to encapsulate your, so I'm assuming a herbal medicine would be some kind of an API that you have extracted from a natural product, and yes, that can be encapsulated within a nano carrier material, and that kind of conjugation or chemistries can happen within these microfluidic devices. I hope that answered your question.
Sorry, I think I was on mute. The question is from AEPL about other what are the other such devices in the market and what is the USP of this device?
So currently we have different kinds of microfluidic platforms in the market, and most of these platforms are imported. They work on the same principle, but the microfluidic chip that they provide, that is a single use chip, and the ones that we provide are reusable, which means that you can use our microfluidic chips for about 50 formulation runs. And added advantage is that the operating cost of the system would be quite less in terms of the consumables that are used. And we are about 40% cost effective, 40% lesser in cost in compared to the imported systems. And primarily that comes in the operating cost of the system. So one time consumables are good enough to use the system for quite a lot of formulation runs.
Right, I think that's a wonderful feature because I think consumables can be a big burden. So that's really a useful feature. We have one more question about from Dr. Somnath, if it can be used for nanopolymers.
So the material of the chip, the material of construction of the microfluidic chip is compatible with a wide range of solvents. So it's compatible with acetone, it's compatible with DCM, ethyl acetate, ethanol. So it largely depends on the type of solvents that your polymer is solubilizing in. And if those solvents are compatible with the material of the chip, then you can use the chip for preparation of any kinds of nanopolymers or nano particles. So that actually depends on the solvent compatibility.
Uh, I think there are many questions. Maybe what we can do is we can ask the participants to raise their hands, and I'll unmute the
Yeah.
participants for any questions. We also have a question from MD Danish about the maximum and minimum size of nanoparticles that can be obtained, size range.
So the microfluidic device that we have, it has a channel dimension of about 300 microns. So this is quite a bigger channel in comparison to your traditional droplet-based devices. So due to this reason, the material, so the nanoparticles ranging from about 50 micron to even micron-sized particles as large as 100 microns can be prepared on the same device. So that's a quite huge range of particle sizes that can be optimized on the platform.
There are also a couple of questions related to the applications. One of them is about can this be used for DNA encapsulation, like for gene delivery, and also about whether there are any other applications of the technology for apart from vaccine development.
Yeah, so primarily the device is used for encapsulation of nucleic acids such as DNA, mRNA, and we have also tested the device for maintaining RNA integrity. So the device does maintain good RNA integrity. There is no degradation happening in terms of nucleic acids because there could be a chance that due to shear forces there could be some degradation, but we have tested the instrument and it has done well in that aspect. So yes, nucleic acids such as DNA, RNA can be encapsulated on the system. Apart from this, for other extended applications, we it is countless because this is essentially a mixing technology. So if you can prepare different kinds of carrier materials such as inorganic materials, we've also prepared zinc oxide nanoparticles, metal nanoparticles, polymeric nanoparticles, even micron sized particles. So the applications could be towards not just vaccines but also any kind of drug delivery system that is used for applications even in other kinds, not just drug delivery, maybe in diagnostic kits as well or theranostics as well. So the applications is endless because the technology is quite simple in itself. It is giving you particles of controlled size and scalability as well as good reproducibility. So that can be extended to any domain as per your area of expertise or research.
Right. And there's also a question about if you're providing services to, like, for example, I think you can provide service for academic projects to your company, and if you can probably maybe process the samples. I think that's the question.
Yeah, I mean, we'd be really happy to do that because we get to also extend an application. So you might be working on different projects, and if that could be an added application of the instrument, we would be happy to host you. And you can perform your optimizations at our facility, or else you can send your samples and we can do the same. And yeah, so we are open for services, and yeah, there is a lot of collaborations that can happen.
Okay, wonderful. There's also a question from Malvika about if you can elaborate on how it can be used in diagnostic kits.
So, some of the diagnostic kits do have nanoparticles in them. These nanoparticles are usually tagged with some kind of an antibody, and So there are some applications. So the gold nanoparticles can be prepared. We also have projects where people are preparing nanoparticles with other kinds of metals that are not patented as of yet. So there are multiple applications in that domain. And like this, your lateral flow kits are also working on the same fabrication principles, so they also use the same kind of materials of constructions. So there could be so currently what we do is more in the formulation space, but if there is any customized design that you require or any customized service, then that can also be explored.
Wonderful. So I think the tech because the technology is emerging, I think a lot of new applications are also in the way, and the fact that uh, Amar Biosystems is inviting you, encouraging all the academic researchers to try out different applications, and definitely more. I think more can come up on the usefulness of the product. And we have one more question about how Teflon-based microfluidic device is developed. I think the question is related to the advantages or features of Teflon-based materials.
So at our facility, we micro machine those micro channels on Teflon based chips, and we the material is quite compatible with a variety of solvents, is what we have noticed. And Teflon being a non-sticky material, it is excellent for use with highly viscous systems. So we have optimized microsphere preparation on these Teflon chips. Now, microspheres are prepared at a quite high concentration of polymers. So imagine 100 mg per ml of your polymer concentration. So this material does not stick on Teflon, and this actually prevents microchannel clogging, and it was actually helpful for us in optimizing formulations on that extreme viscosities, as well as if you're using sticky surfactants like PVA that tend to precipitate inside the channels. So having Teflon based chips sort of circumvents that problem.
Correct.
And it's also comparatively cheaper than glass based chips, because a glass chip, once you order and it clogs, the channel clogs, it's practically impossible to clean that and reuse it unless you use extremely corrosive materials to clean the chip, and that can also damage the microchannels within the chip. But Teflon based chip, it's easier to clean with your solvents, and in our case our chips are actually bonded physically, so there's no chemical bonding happening, so we can actually open the chip as well in case it clogs.
All
So
right.
these are some advantages with the chip.
So thank you so much, Iti, for this wonderful session. And we'll put out the contact information in the chat box so that the participants can reach out to the team at Amar Biosystems if you have any queries regarding the technology as well as the company. So
Sure.
thank you so much, Kirti. Uh, Now we are going to have a special session, a special lecture by Dr. Swetha Raghavan uh, on mRNA vaccines and interventions for dengue.